Thanks, everybody, for the invitation to this series of meetings. We are very happy to share with you these new tools that we have just recently open sourced, this OVO application, or better to say ecosystem for de novo protein design. So I'm Marco, and today, together with Teresa and David, we will present the coolest features, let's say, of OVO. So maybe just a few words about our team. We are the research and innovation team here in MSD in Prague. These are a collection of photos of some of the members of our team, basically all of the people that have contributed to develop this ecosystem. And we historically have published many papers and open sourced many tools, actually, in the past. We've been focused on the antibody, for instance, space, but recently, in the last few years, we are kind of shifting our focus on the de novo design, as it is one of the most promising, I would say, fields for protein design in general. So you are free to check our preprint on the bioarchive at this link. And I will start with a slide about the motivation that led us to develop this tool. So it was almost 2 years ago that we started to envision this ecosystem. And the main motivation was that, especially at that time, it was very difficult to reproduce the, let's say, gold standards for de novo design. So, of course, the tools were very promising, and the papers were showing us very outstanding, I'd say, results. But it was very difficult to reproduce that work, and also the internal effort that we had in the company to try to leverage such technologies. Of course, the field was rapidly improving, and other open source tools were popping up in the community, and it was very difficult to keep up the pace there, and also to enclose them in a single, let's say, infrastructure to have a good benchmark. And this led to a lot of duplication of work, as we have already heard in the introduction. So in the end, we started to work on this tool, trying to hit some targets, which is the tool needed to be easy to use. So that's why the user interface and the pipeline automation, shareable to basically facilitate the knowledge transfer within any organization. So linkable and downloadable designs and reports. And also the storage standardization, which is a very important point here. And other advanced options, such as the plugin tools and the command line interface that we will describe later on in the presentation. So this is a very, it's a schematics of the core. Of course, OVO, it's built around the main state-of-the-art pipeline for de novo design. So you can recognize the RF diffusion end-to-end pipeline and the Bindcraft. We added, in that sense, one internal pipeline built entirely by ourselves, which is ProteinQC. It used to be a separate application, actually, but we integrated it in Novo to have it as the final step of the whole pipeline, so that we have a unique integrated end-to-end pipeline from the backbone generation to the filtered designs. And as you can see here, the typical use cases, so protein scaffolding, binder design, binder diversification, that we are currently trying to including in our pipeline, in our application. But it's important also to say that this ecosystem was built by us for two important reasons. Trying to democratize this tool in the community, in the open source community. So trying to facilitate basically the usage of this application for the open source community, and also to make it flexible enough to be usable for different purposes, which means that if some teams are very strong or have very established protocols for the lab protein design, but they don't have the capabilities of developing their own in silico pipeline, OVO provides the easy user interface and the automated pipeline to basically kill the overhead needed to implement these tools by yourselves. But also, as we will see later, for the most expert users, so the most computational people, they have the possibility of using this whole framework to include their own custom pipelines or to modify the existing pipelines to improve them in the same exact framework. And this would help benchmarking, comparison, even sharing within the organization. So let's start with the first step. So when you first encounter Ovo, so of course, we encourage you to check on the documentation at this link. So we try to be very detailed there and try to explain as much as possible what are the first steps and what are the features. I have here a very quick schema of how you could start to configure Ovo in your machine. So Ovo is pip installable. The entire Ovo can be installed in your machine with a single installation. And afterwards, the few command line commands that you have to use in order to, for the first configuration is the Ovo init. Ovo init home allows you to establish the database, which is one of the database and the storage, which is where we basically store the metadata of the designs and the descriptors for the designs, and the storage where we have basically, we decide where to put all our input and output designs, so the heavy files of the pipeline. And then there are the other three pillars of the configuration, let's say the working directory, where the pipelines are dropping all the intermediate files, the configuration file, where you will basically annotate all the important settings for your Ovo to run in the machine, and the reference files, which are more or less all the files that are needed for the pipelines. So here I put an example, which is RF diffusion weights, for instance, are downloaded and stored in the configuration directory Ovo. There are several Ovo init commands. Some of them, for instance, will allow you to test the main features of the application. In this case, the Ovo init preview will run for the first time the RF diffusion pipeline, for instance, and will help to set up the scheduler. As we will see later, Ovo supports different schedulers, namely PBS, Slurm HPC, AWS Cloud, and also local scheduler for running it on your own laptop. So this is probably most technical slide here, and it is the core orchestrator of Ovo pipelines, which is Nextflow, a very well established tool in the bioinformatics community. The Nextflow basically integrates seamlessly all the processes associated in the Ovo pipelines, in the Novo pipelines, and allows for grid jobs, parallelization, even automated retries. It supports different container set package manager, and basically queues up all your jobs in a very efficient way. So how the pipeline starts? Usually it starts with some input, user-defined inputs from the Ovo web application, for instance. Then each pipeline is divided into processes. So these tasks are basically backbone generation, let's say 100 of binders, generated binders, then in the backbone. Then you have as a second task maybe the generation of the sequence, as a third task some backbone or sidechain metrics for early filtering. Then you want to basically predict, maybe with AlphaFold or so some designability test. So all these processes are basically run subsequently by Nextflow, as I was saying, in a seamless way. And the output of that are stored in a predefined configured folder. And if you are in the web app, the status are checked continuously. Afterwards, at the end of the process execution, all the outputs, relevant files are stored in this work direct set dir, uniquely identified by the job ID, and the output are processed and saved in the Ovo storage. Together with the Ovo storage, the descriptors that are calculated in ProteinQC associated with each design, as well as the design metadata, are stored in the database. And just I want to stress that the database and the OVO storage are the single database and the OVO storage that you set up in your local machine or in your server by yourself, and this can be shared amongst all the team members. So it's something that really speed up these let's say, this overhead that usually we have in different organizations, both academical and in company organization. Each of these tasks are run in containers. So these containers are basically package managers and are completely isolated, and they help basically with the dependency management, as I was saying. So Conda, Docker, and Singularity, they can be installed and downloaded from the documentation. Finally, this is an example of the configuration file, which is basically a YML file. You can see here that you can easily set up the scheduler. You can use the Slurm scheduler, for instance, to run your jobs or your pipelines in the HPC, as well as the local scheduler. And on the Nextflow pipeline will take this config file, the Nextflow Slurm Singularity, and will process your tasks with a defined specified variable, such as using GPUs, CPUs, or particular load of memories. This is the final slide from a bit, and this is just a way to show you how all the the moving parts of OVO are interacting to give you this full ecosystem. It's important to see that the command line interface and the Python interface also allows you to use OVO exactly as any typical Python package. So you import it and you use all the functions of OVO in your Jupyter notebook to further analyze your design downstream. And after that, I will pass words over to Teresa.
Okay, so I will transition into a quick demo of the user interface. So just what you will see is that we support basically 3 different pipelines for RF diffusion, and that's the protein scaffold design, protein binder design, and binder diversification. And then we also have a
I don't know about other people, but I think I'm still seeing the old slides. Did you share the screen?
Yeah,
that's correct.
That's fine. So we will transition. And then we also have Minecraft, but we are not gonna show that. So we can move to another slide. So that's how the UI looks like. You see that you have a project, and then for scaffold design, you land on an introduction page where you have the description of the workflow and you input the input structure. Then for scaffold design, you mark the fixed segments. So you can either do that in the sequence or by selecting directly in the 3D structure. So we are using Molstar for the structure visualization, and we also support different surface or coloring visualization. So here's the hydrophobicity and molecular surface visualization. And then in the next step, you're gonna form the context, which is basically the input for RF diffusion. So you have this drag and drop context editor, where you specify how you want to link the selected segments from the previous steps. So that can be a single value or can be a range. And on the right side, you can see basically in the 3D structure, the selected segments from the previous steps. And optionally, you can generate a quick preview of just few iterations of RF diffusion. The next step is optional, that's sequence inpainting, so selecting residues from the fixed segments that will be redesigned. And lastly, you just select your project round, so you can have multiple rounds in your project. You name the pool of designs such that you remember or you have some description of it. And then you set the number of backbones and number of sequences that you want to generate, and then there are additional settings. And now we can move on to the second workflow, which is binder design, and the interface is very similar. So you again have a description of the workflow, and then in the input you can set the PDB file. You can actually also reuse your previous runs if you just want to submit the same workflow with more designs. And for binder design, you're going to select the chain of interest, and then you can optionally select the hotspots. So again, you can select them from the sequence or from the structure directly. And the next step is optional. That's trimming of the target chain. So to reduce the computation time and cost, you can trim the original structure to shorter parts, so the relevant domain. And you can see the side-by-side view before and after trimming for what you have selected as the range. Then in the next step, we're going to set the binder length, so that can be a single value or a range. And you can also set again the hotspot residue. So if you haven't done that in the structure or sequence mode at the beginning, you can do it here. And then you can again submit. So you select the project round and you name the pool of your designs and you give it a description. You set the number of structure designs and the number of sequence designs. You can choose between ligand MPNN and protein MPNN fast relax for the sequence design. And you can set advanced parameters. So those are for arc diffusion and MPNN. You can also set different tests for refolding and set the thresholds for acceptance. So that can also be configured later. And finally, you review the parameters and you submit. So after the submission, you will be redirected to jobs page where you can monitor the status of your job. And once the job is finished, you can open the details page where you will see how many of the designs are passing the acceptance thresholds. So we have some acceptance thresholds by default, but you can change them. You can disable and enable different criteria and change the thresholds. And below that, you can browse the designs that are passing those thresholds. So this is great because you don't need to move the files to your favorite structure viewer, but you can do it directly in Novo. So you see the results also of the refolding and the MPNN design and the arc diffusion backbones. And in the second view, you see all designs. So this is the typical AlphaFold PAE versus RMSD. And the plot is interactive, so you can select a section of the plot and explore the design. So those are designs that are not so successful with AlphaFold, but here in this part we can explore the more successful designs. So you can see also some basic metrics, but for the metrics we then have a separate view. So here it's rather about the visualizations, and you can also download all the corresponding files. So then once you have filtered your designs, you can navigate to designs page where you have a couple of views. The first one is Explorer and the interface is similar as before. So you have this interactive scatter plot and you can decide different descriptors for the axes. You can browse the structures of the designs. So you see again the arc diffusion backbone design and the fast relax and also the results of the refolding. And then, the Demostar also allows for some more advanced visualizations. So we can also move it to full screen and explore. And then the second view is ProteinQC, which allows you to submit additional computation of descriptors for your accepted designs. And we put them into context of reference distribution. So that's the gray distribution above, and below you see the distribution of your designs. If you click on show more, you can browse the designs. We also give them flags, so you can be notified if there are outliers in your data or designs with some suspicious values of those descriptors. And we also show both some sequence and structure-based descriptors such as predicted solubility or hydrophobic patches. We also display some basic backbone metrics and refolding. And lastly, you can also look at this interface analyzer view, where you see some basic overviews for the binders, such as the target interface residues. And you can also filter your designs by the target interface residues, so you can only see result designs that are having this particular residue, and some basic overview. And lastly, you can also submit additional refolding. So you can submit AlphaFold monomer, multimer with template or without. And that's all for the binder design. And the last workflow is binder diversification. So here you again have the description of the workflow, and you can input either PDB file, or you can use OVO ID of your previous OVO designs, and you can submit partial diffusion. So here you can select the residues that you want to be redesigned. So you can select again from the sequence or directly from the structure. You can generate a preview of few iterations, and then similar as before, you select the project route, name the pool of your designs, and set the number of backbones and number of sequences, set your sequence design method, and also you can pass advanced settings. So those can be, for example, backbone filtering. So if you want, if you don't want helical binders, you can pass this filter. And then again, you submit and wait for your job to finish. So that can be overnight, and in the morning, you just open OVO and you will see that the job has finished.
Thanks, Teresa. So I will continue showing here first the other way of interacting with OVO is through the Python API. This is a way of submitting the workflows and processing the results and looking at the descriptors. So here is a way how you instantiate a new workflow submission. So you just pass these parameters, then you call this submit function, and then you call this process results function that waits for the job to finish. And then once it's finished, we'll store the results from the output folders to the database. And once it's in the database, you can use this DB object to interact with all the data, so the designs, descriptors. So you can fetch, for example, them as a data frame and then just visualize however you like. And it's also commonly how we do our final filtering is kind of to continue on the reproducibility, to do some filtering on the accepted designs in OVO, and then do some downstream filtering in Jupyter by interacting directly with the OVO database. Another thing that we wanted to share that we're really excited about is plugins. So what OVO allows is to extend the pipelines and also the web application by independent Python packages. So what that means for the user is that hopefully in the future we will get this momentum to create different plugins. So this means that, for instance, one plugin that we published was for prompt, which is our open source method to evaluate humanness of proteins. This can be used on antibodies, but also de novo designed proteins. So once the user pip installs this separate package, actually they will see a new tab appear in the OVO application that enables running more workflows to annotate designs. In this case, measure the humanness, so the human peptide content percentage. Another plugin we are working on in collaboration with the ProteinDJ authors is for ProteinDJ, which is another Nextflow pipeline. So this would enable you to, if you're running ProteinDJ already, to kind of import your results into OVO, but also to submit the ProteinDJ pipeline from OVO directly. So this is where we would really love to see the collaboration happen, is that this is actually a completely independent way of contributing. So you don't have to like submit a pull request for our repo. This is your own repo. So it's a Python package that needs to contain some logic for the UI. This can actually just be a single Python script, thanks to Streamlit, which is very easy to use. And then the pipeline, which is Nextflow. So this could even wrap a single script. If you have a single pipeline that could run on a single machine, it should be very simple to create, but you can also use it to create very complicated workflows that orchestrate the individual tasks. So to summarize the strengths, we think that The main strength is the ease of installation and ease of use. So focusing really on the users and the kind of reproducibility aspect and broadening the user base. So the maybe one thing to mention for the installation is that if you are using Singularity or Aptainer on the cluster, all the containers are pre-built. So it all happens kind of in the background as you run the pipeline, and nothing is really installed. It's all just downloaded, pre-installed. As Marco mentioned, when you set up Ovo, you can set it up for your whole institution, so everyone uses the same database. You can set up the web app as well to interact and share the links, but you can also interact directly with the database independently, and everyone has the same, let's say, ground truth. So for the submission, Nextflow really is the time-tested way to support these different orchestration engines. Actually, it's really, we also use it internally on Slurm, PBS, and AWS. It was very easy to configure. You can really customize how different tasks are running, different queues, and different resource requirements. Thanks to the containers, the dependencies are isolated, and they're also, I would say, future-proof in the sense that the Ovo doesn't declare the only version that needs to be installed of a package. These can be each pipeline can use different version of a container. So then long term, I think this is a sustainable way of using the tools, is that you can have these pre-built containers and then you can just keep adding more. And also the container is independent of the code of the step. For instance, we can have a RF diffusion container, and then you can use RF diffusion different ways for the scaffold design, like motif scaffolding or binder design. So that's flexibility. For plugins, I think what's great is that the development becomes decentralized, so more people can develop it, and they can also just keep the plugin for their internal use. They don't have to publish it. It's a good way of I think building on top of Ovo, but you can also publish externally, of course, what we'd like to see. And finally, I think the shared data model, the fact that everyone has the designs stored the same way in a database should help integrate more tools to enable more downstream analysis compared to just having a folder with PDB files or CSV with metrics. Also wanted to note the limitations. Maybe for some experts, workflow developers, might be frustrating a bit that we focus really on the end-to-end design. It's kind of hard to modify the pipeline code on the fly, especially as a user. You don't modify the code, you just run it as it is. So if you're used to, for instance, running just the backbone design and then run sequence design, with different parameters, there's going to be less flexibility for you. But again, this is a conscious choice that the point is to make the results reproducible and have all the pipeline parameters linked to the final designs in the database. Nextflow, of course, there's some learning curve for people to start using it. And also there are some limitations compared to, for example, ProtFlow, which I think is a really cool alternative, is that Nextflow is not so good in resuming. For instance, if you want to just develop the pipeline as you go, it's typically when you want to resume an existing run, Nextflow is very strict in checking that nothing changed. So if anything changed in your parameters or in your code, it will just rerun the whole pipeline again. So that's really less flexible if you want to have more control. The web app does not really show intermediate results. This might be something that people expect to see. You can still see them, of course, in the output directories, but for the web app, you need to wait until the whole run finishes. But typically, thanks to this batching and parallelization, would be overnight, so it's not so limiting. For the plugins, one might think that you could create a plugin to just switch RF diffusion to RF diffusion 2 in our pipeline, but unfortunately, that's not so simple. The plugin is really to introduce new pipelines. So of course, you could fork our pipeline, modify it a bit, but you cannot really use it to modify existing pipelines. And happy to hear more feedback. I think we would love to send some form after the call to receive some feedback. So keep an eye for that. And just a quick wrap up for the roadmap. So importantly, if you're already installed Ovo, I recommend updating to the latest version where we fixed a bug with our initial guess protocol. So we were actually not enabling initial guess as we thought. We took a code from the RF peptide supplement. So in case you did that, you might want to check your code, because only realized now that we actually were running AlphaFold without the initial guess initialization. Surprisingly, it still gave us good metrics for a lot of the design, so we didn't realize it for a long time. But now with this fix, you should get, like, I would say in our experience, like at least twice more successful designs with the default results. We're working on a few things. I won't spend too much time on it. We're working on bolts-gen integration, also bolts2 for refolding, looking into our diffusion 2. Really happy to discuss that later. And also, we currently use Nextflow only, but it should actually be easy to set up different types of pipelines that all could be like a single place to submit different arbitrary pipelines. So it could just be a bash script on your infrastructure. So all we need is to be able to check the status of that job. So that's something that could come in the future. Yeah, I think I'll wrap it up here. Most importantly, help is welcome. So please see our issues in GitHub and feel free to submit more. There are some already that we marked as good first issue and help wanted. So that would be great to get started. But yeah, happy to hear more. Yeah, thanks again.
Thank you very much. I'll give you a round of applause to everybody. I think this is very impressive and it sounds really cool and really interesting, sort of very spot on. And now we have some time for questions. If you have questions, type them in the chat. If you want to ask them in person, raise your hand. And I'll start with the most asked question, which you touched on briefly, but how difficult would it be to plug in your own blocks inside, right? How, if you want to use bolts instead of AlphaFold, or if you want to use soluble protein MPNN instead of the regular MPNN and stuff like this, would you have to, what does forking the entire pipeline entail?
Yeah, that's a really good question. Sometimes it's more difficult than it might seem, exactly for this reason that the focus is more on reproducibility and standardization, let's say. So we didn't invest that much time to make it easy to fork the pipeline, but definitely would be nice to pilot test this. Some things might be easier than others, like soluble MPNN should just be a very quick. Actually, we could add a GitHub issue if anyone is willing to contribute that. It should be easy. We already let you select the RF diffusion weights, so we could do the same for ligand MPNN. Plugging a different method then becomes kind of a design choice that we would need to have a standard way of outputting the backbone designs. I know ProteinDJ has different options that are maybe more plug and play, that they have this kind of JSON format to extract this metadata. So yeah, I think
how are things stored in the database? Because you mentioned a sort of an intermediate representation. Is that like a Rosetta pose, or is it just like a single record describing the entire design? So
you mean
maybe if
the pipeline?
Yeah, throughout the pipeline, right?
So for our pipeline, we use there's no like overarching step. It's more like between each two steps. So each step can take inputs from another. Like let's say MPNN takes us the input from RF diffusion. So then you just kind of write the logic there to convert the files and prepare them for MPNN. So the MPNN step takes the TRB files. Well, actually, what we do in the RF diffusion step already is that we add a header to the PDB file where we specify some metadata, and then we read that metadata in the MPNN step. So then there's no, like, compared to ProtFlow, it's like less managed on like the global level. It's more like the individual tasks need to prepare their own input based on the previous task.
That's why it's more work to switch blocks. Very cool. So there's another question. Is there a limitation on the size of the protein that can be designed? I think that's probably limited by your hardware and in general by, the programs themselves. Do you have some plugin options for antibody workflows?
Yeah, that would be a cool addition. I think there you can already upload So we had that example in the paper that you can upload structures, antibody structures, and then analyze them with ProteinQC. So that could be one use case to run, let's say, positive surface patch analysis or hydrophobic patch analysis. So you can do that already. We are planning to add sequence-based design. So you could just upload these sequences, and then once we have that, it would be cool to have plugins for, let's say, a bodybuilder to run structure prediction. So we don't plan on working on that because we have other tools internally that we are using for that. But if anyone is gonna join that we would be happy to see contributions in that space.
And you also mentioned that you wanted to organize like separate plugin hands-on tutorial. Is that still
Yeah.
an
I think it
option?
would be nice to have a maybe more technical overview of the Nextflow pipelines and discussing, showing a really simple way of building the simplest plugin imaginable, maybe. And then really, I would really love to hear people's thoughts on what they would love to do with plugins, and then see how we can make that easier. So I know that people want RVision 2. That's one thing. So one way also would be that doesn't have to be a plugin. We could just extend our existing pipeline. But it would be nice to have an example of like how to fork the pipeline and
I think this will become clearer also in the other presentations, because each pipeline each workflow sort of solves a specific subset of problems. So I think that's we'll get an overview in the next few weeks of that as well. And another question is, how easy is it to add metrics to the ProteinQC? I don't know if you want to count 4 alanines in a row or something like that.
I would be happy to discuss that. I think we could also have separate plugins. So that's I think the easiest use case for the plugin is to have these descriptor annotations. And there's also a nice example with this prompt package, which does humanness annotation. I will share the link later in the chat for all these. So that just lets you run this single pip installable tool and show it in the UI. So that could be also another way to do that. But yeah, happy if you want to submit an issue and a pull request, we can add that to the ProteinQC as well.
Cool. Perfect. People are also asking, this is a little bit out of scope, but people are also asking if you have any idea which metrics are most correlated with experimental success.
We'll know more on that. I don't know if there'll be a publication, but for now we tried to set the defaults as best as we could, but this was really consistent with the literature, so we don't have any new insights yet to share.
Yeah. Perfect. Let's give the presenters another round of virtual applause.